I'm really delighted to introduce today's speaker. A couple of years ago, Janet Holbrook and I were asked to serve on an advisory committee to the Institute for Clinical and Translational Research at Johns Hopkins All Children's Hospital in Florida. And that's where we met Neil Goldenberg. And we quickly discovered that he was a clinical trialist and that he had been doing thrombosis trials for a long time and that he was running their clinical trials unit. We decided, well, we really need to have him come speak and talk to us about what he's doing. And that's led to this talk. Neil is a graduate of McGill University. He got his MD, his PhD at the University of Colorado. He did a residency in pediatrics at the University of South Florida and a hematology oncology fellowship at the University of Colorado. He then joined the University of Colorado faculty, and in 2012 moved to Johns Hopkins, where he's now a professor of both pediatrics and medicine. He's the Associate Dean for Clinical Research at the Johns Hopkins All Children's Hospital in Florida, and he's the director of the Johns Hopkins All Children's Institute for Clinical and Translational Research. His area of expertise is pediatric thrombosis, and he has chaired clinical trials in this, and in particular, he's chaired one incredibly large multi-center trial with over 50 clinical centers on duration of anticoagulation treatment for children who've had thromboembolic events. He's going to talk to us about, here's his title, Design, Implementation, and Oversight of Multi-Center Trials in Pediatric Venous Thromboembolism Treatment and Prevention. And we look forward to his talk today. Neil.
Thanks so much, Doug. It's a pleasure to be here with you, and I look forward to attending many more of these seminar series and also having more faculty from Florida attend and be engaged. I was saying we have a small but passionate group of pediatric clinical trialists, or many of them early career, and this would be a great forum for them to interact with their colleagues on the main campus in Baltimore. So I'm gonna have Taylor, if you don't mind, go to the next slide. And in a moment, I'm gonna just address the elephant in the room, which is, so kids actually get venous thromboembolism, right? And tell us more about this before we talk about the trial. Another, I guess, pseudonym for this talk is doing multi-center trials in challenging disease settings. Pediatric venous thromboembolism doesn't quite qualify as a rare disease in terms of grant mechanisms, et cetera, but it is certainly a low frequency disease. And so the other challenge would be trying to complete a trial during a pandemic. I'm not sure how much we'll get into that, but I will try to leave plenty of time at the end for interactive Q&A. Next slide, please, Taylor. Some disclosures. I get some research support for one of the trials discussed here through a U01 from NHLBI and some salary support. I serve as a pediatric population liaison for the CTSA. And then I'm involved in a number of industry-sponsored clinical trials at an oversight committee level, particularly data and safety monitoring committees or steering committees. And it's one of my passions that, we have a way for academicians who are trialists to help inform the design of the industry-sponsored trials that ultimately lead to treatments, especially for kids. Next slide. So I'm going to talk a little bit about the KidsDot trial and the COVAC-TP trial. The latter was a trial we launched during the pandemic in response to blood clotting complications that were being seen not only in adults, but in kids. We focused on the pediatric challenge. And then a little bit of the impetus for the design and really tailoring the study design to the state of evidence in this area where evidence for a low frequency disease in kids was not incredibly robust when we started this effort. And make a little pitch for considering a pilot RCT design stage before launching a very large definitive RCT when you're challenged by certain feasibility concerns for which there's not a lot of evidence prior to launching that larger trial. I'm going to really focus on number 3, which is the implementation aspects, utilizing institutional infrastructure, having a DCC as well as a clinical coordinating center to really execute a large study. Often we don't talk a lot, at least in our, our teaching and training around committee roles. And so I'll spend just a few minutes on clinical endpoint adjudication committee, DSMB, and steering committee, and a little bit about their functions. And hopefully at least the students and junior folks may find that helpful, as they're not often really in textbooks or other materials in our curriculum. And then lastly, give you a glimpse of some of the recently published findings in JAMA for KidsDOT, and then for COVAC TP, some of the findings that are under review for publication. And then hopefully a lot of time for Q&A, as we said. Next slide. So I'm going to take a minute to talk about venous thromboembolism in children before I give you the who, what, when, where, why, and how about KidsDOT trial. And so venous thromboembolism occurs in approximately 1 in 200 hospitalized children. If you look at children who are critically ill in the hospital, it's approximately 1 in 50 children will develop a venous thromboembolic event. And so as you get further into risk stratification, there is a role for developing evidence for prevention of VTE, particularly in the hospitalized child. And it's not unique to adolescents. The entire age spectrum is affected. And in fact, one of the peaks in incidence for pediatric VTE is the neonatal period. Those are often not healthy babies, but babies who have other conditions, particularly premature infants who then have to be in the hospital, often have catheters in a large vessel, et cetera. But it's interesting that in the otherwise healthy child in the community setting outside of the hospital, the incidence is about 1 in 10,000. So, depending on which way you look at it, it may be a low frequency, moderate frequency, or even rare condition. So one of the challenges is that the knowledge of this being a problem in kids, of course, lags behind the knowledge of it being an important issue in older adults. And so our evidence is behind. With evidence on how long to actually treat a child to prevent a recurrence and hopefully avoid bleeding complications. That evidence on how long an acute VTE should be treated in a child is completely unsupported by high quality evidence, whereas in adults, those pivotal trials were done in the early 90s. So you get a sense of how long, we're talking 3 decades of lag to understand basics about how to treat a clot in a child. That said, the tradition in pediatric medicine is often a cut and paste of the adult evidence, and then at some point pursue hopefully definitive evidence that would show whether children need to be treated differently or not. So that's kind of the background for the KidsDOT trial. When I was a fellow in Colorado, as Doug mentioned, I was fortunate to be able to do a prospective cohort study to try to define some prognostic factors in VTE. And we didn't really have any good quality data on that. But through that work, which by some fluke was published in New England Journal, we started in our pediatric VTE field to understand that you've got some kids who are low risk and some kids who are high risk for adverse outcomes. And KidsDOT was targeting the low risk group. So we looked at children under, and we defined that using the NICHD definition of 21 years of age, not an FDA type of definition as under 18, who had a provoked VTE event. And that provoked is important because we know from the adult literature and from pediatric experience before we launched this trial that if you have a clot completely out of the blue, no clear provoking factor that was reversible in nature, then we think you are at high risk for recurrent events. We couldn't find out, what caused it, and therefore, can we ever get you off blood thinner treatment, right? Whereas provoked VTE, we know has a lower risk for recurrence, particularly when the provoking factor has been mitigated or has resolved, and then we feel like you can have a finite duration of anticoagulation. So in part of making sure we had the right low-risk population for this trial, it was kids with provoked VTE, which constitutes somewhere between 90 to 95% of VTE in kids. So it's still the vast majority of children by selecting provoked. It was an RCT, randomized controlled trial on duration of anticoagulation. So we're not focused on a particular drug. It was clinically prescribed anticoagulation. We were focused on how long. So the convention was 3 months, the cut and paste from the adult evidence for provoked VTE. And based on some of our experiences and our hypothesis that kids have a shorter period of increased risk for recurrent events after an initial event, we thought that cutting the duration in half to 6 weeks would be at least as safe, but as effective as the conventional 3 months. And we say at least it's safe because you're exposing someone to a blood thinner, quote unquote, an anti-clotting medication for half the time you would be exposing them under the conventional standard of care. So this started in 2004 officially with its first grant for planning grant purposes, and then under a K-23 from 2007 to 2013, and then a bridge grant. It underwent a pre-specified pilot feasibility component where we took a bet and a gamble that if our assumptions, some of our key assumptions for the definitive RCT were validated during the pilot phase, that we could seamlessly continue to enroll patient number 101 and beyond in the trial. Had we not been able to substantiate our assumptions, we would have had to start back at patient 0. But one of the things I will say is I would not probably want to do a large RCT again under a career development award like a K-23, because those of you who have been involved in those know that you really can only propose like 4 centers at max in your trial. And so you have to have creative ways of how can I bring on other centers without really slowing down my trial. This was ultimately, after the K-23 period, conducted at 51 centers, 42 of whom actually enrolled patients on the randomized trial. The point of it, the why, was to inform the standard of care on duration in these kids. And the how is a little bit interesting, but I don't think we'll have time to go into it now. But if you look into the literature, there's 1 or 2 other examples of something called a parallel cohort RCT. One of the first ones that I could find was antihypertensive trial in adults. But the point here was, I mentioned this is low frequency disease. In our trial, we wanted to make sure that we didn't randomize kids at 6 weeks to stop treatment versus to continue for the full 3 months if they had some features that we didn't really have equipoise to feel like they were going to have favorable outcome and be low risk for recurrent clots. And so if a child at the end of 6 weeks had repeat imaging of their deep vein thrombosis, and it was completely occluding blood flow in that venous segment, then we felt those kids should not be randomized. And so that was one of the randomization criteria. And those kids remained on study, but were not randomized if they had complete occlusion at 6 weeks. And so that's the parallel cohort part of the parallel cohort RCT. And why do that? Well, if you're going to get 51 centers together and you're going to work on getting all these grants, and you've got so little evidence in your field from multi-center, observational studies, much less RCTs, why throw those patients out? Why not follow them in a very standardized fashion in the same way you do the RCT patients and get really good natural history data that could inform the next interventional trial in that subpopulation? So that was the impetus for the parallel cohort RCT. I think everybody's familiar with prospective randomized open label blinded endpoint. So I won't talk about that. And the hypothesis was non-inferiority. Next slide. And I promise I won't take so much time on every other slide, but I wanted to get into some of the operational aspects even at the intro stage. So COVAC TP, we launched in June 2020 during the pandemic as a single arm phase 2 trial of the low molecular weight heparin, enoxaparin, given every 12 hours subcutaneously and dose adjusted to achieve a target pharmacodynamic level that in the standard of care uses something called the anti-factor 10A activity with a target of 0.2 to 0.4 units, activity units per ml on a lab test performed 4 hours after a dose. And we conducted this at 18 centers nationally. I can't recall exactly how many of those ended up enrolling patients, but it was not all 18. And the purpose was primarily to evaluate safety with some a priori definitions of frequency of clinically relevant bleeding as the safety endpoint. Secondarily, we wanted to look at dose finding by age group to achieve the targeted activity levels in plasma. And our tertiary or exploratory endpoint was to get an early glimpse of efficacy. How many kids were still getting thromboembolic events despite having low molecular weight heparin prophylaxis when they are hospitalized for COVID-19. So we'll go to the next slide. So in designing the trial, I think it is really important, and I won't belabor the point, to have insight to where the gap in knowledge is and what's the state of evidence in the literature. And no sooner do you put up a slide like this than you start to deconstruct it and say, well, that classic, textbook preclinical to phase 1, phase 2, phase 3, it doesn't always occur, especially, in rare diseases. Sometimes you have more creative approaches to getting through those hoops of evidence while still minimizing, exposure and safety concerns to the patients as you go forward in each phase. But nevertheless, acknowledging all the caveats about proceeding in this kind of fashion from top row to bottom row, it's important, I think, especially for early career folks, to not immediately say, oh, I'm going to do a randomized controlled trial. The next step often in our training programs here with residents, fellows, and junior faculty is to say, well, actually, no, you need to do a prospective cohort study at a single institution, and then you want to validate your assumptions in a multicenter cohort study. And then you can start proceeding into your phase 2 in kids, and then maybe a phase 3, et cetera. So I don't want to overgeneralize because then that will start to fall apart. But I think it's an important question, and we were deliberate about that in each of the 2 trials I just mentioned. Next slide. So I am going to skip this other than to say that I do think there is a role for a pilot RCT to be done before the definitive RCT to validate assumptions that could make or break your RCT. And particularly, what are your outcome rates for your primary endpoints? If your data are not solid on that, or if the data you have didn't use a standardized definition that is now the international standard in your field, you ought to validate those assumptions before you have your definitively powered trial. If it wasn't in the specific target population of your trial, again, your outcome frequencies from the literature may not be reliable. And as you all know, much more than some of my clinical investigator colleagues, that can make or break the study design based on these assumptions. And the other assumption that's really important is your recruitment, randomization, and retention rates. And we're way often too optimistic about those. And so it can be very important to validate that with your actual protocol. And so just a pitch for doing pilot RCTs more and not having the pilot RCT look the same as a definitive RCT. The aims are different. Aim is to have as precise as possible an estimate of recruitment, retention, randomization, as precise as possible, an estimate of the outcome, but you're not trying to compare in that pilot. All right, next slide. So we actually undertook to do that for the first 100 patients. We pre-specified this in the protocol. We pre-specified aims that were different than the non-inferiority aims of the RCT, and we published it in 2015, right after completing the K23. Next slide. And so I just wanted, in terms of the point about how to operationalize large RCTs in a challenging setting or challenging population, is, too often early career folks think of, I have a one-to-one correspondence between my grant and my project. Something like this takes multiple grants, sometimes over multiple phases. And so I'm just listing most of the ones involved, and then in red are the NIH kind of K and U phases. Next slide. So infrastructure is really critical, and it's wonderful when institutions have had a very forward-thinking approach to supporting multi-center trialists by establishing key infrastructure for and expertise for the design and for the execution, oversight, and analysis of a trial. And in fact, the infrastructure we've tried to build on the Johns Hopkins All Children's campus is to support multi-center trialists, not just site-based participation in someone else's trial. Both are really critical, but we wanted to really attract, over the years to come, more and more Hopkins faculty who are conducting multi-center studies. Next slide. So one model involves somewhat borrowing from an academic research organization that runs multi-center studies, borrowing from that model and trying to building it on your campus. If you're a children's hospital like us, making sure that you have not just your site-based participatory infrastructure, but the ability to coordinate site participation across a multi-center trial, or to coordinate the database design, data management, and analysis, planning and execution. And so I won't belabor DCC. Folks like Dr. Jabs and others have far more expertise than me in DCC, but we have a small and growing DCC focused on pediatrics here. It was not used for KidsDOT, but it was used for the COVAC TP study. And the DCC here is involved more in the secondary manuscripts coming from KidsDOT. In an RCT, it's important when you're having blinded endpoints, et cetera, to separate your DCC from your CCC so that you minimize investigator bias in the analyses. And so NIH actually had, NHLBI had 2 U01s that were a mechanism to support multi-center trial applications. And so we had a U01 based in Colorado for the DCC and a U01 based at Hopkins on the Florida campus for the Clinical Coordinating Center. Next slide. And so, what is the infrastructure look like at a Clinical Coordinating Center? You have a research operations group focused on project management for multi-center multi-site coordination, helping to assure that the protocol and informed consent documents are all consistent. This preceded the SIRB mandate and infrastructure. Now, a lot of this can rely on SIRB kind of centralized support. But still engaging sites, qualifying sites in terms of their experience, qualifying investigators in terms of their experience, et cetera, is all a centralized function that a CCC can provide. For a study that's under an IND or an IDE, having a support core to help an investigator to do the FDA submissions and the updates, as well as the annual reports, is very helpful. We have a small support core for that here, and there's great communication also with the main campus ICTR's IDS group. And then we have tried, and I always encourage early career folks to build in parallel biospecimen banking components to their clinical trials whenever feasible, because often there's the opportunity for biologic markers to help with further risk stratification in a disease. So one of the early infrastructure building efforts we did on this campus was a state-of-the-art pediatric focused biorepository that became College of American Pathologists accredited to support these studies. So central biobanking function, in other words, making kits for specimen collection, distributing those kits like you would an investigational drug to all of your participating sites, having your lab manual procedures for processing, and then shipping back to your central biorepository, logging those in and maintaining them until time to distribute for the translational aims is a function that's really helpful. And so we built that as part of the CCC. The Data Coordinating Center, I won't go into the details because I think others can speak to that far more eloquently and with greater expertise, but obviously having database design, infrastructure, data management, data quality assurance, epi biostats, and in particular, as we'll talk about in a moment, committee coordination. So we've envisioned steering committee management as part of the CCC function with charter templates and SOPs around managing a steering committee, maintaining minutes, et cetera, providing reports from Data and Safety Monitoring Board to the steering committee, et cetera. But that the Data and Safety Monitoring Board management lives at the Data Coordinating Center level, unless the sponsor, such as NIH, is actually managing the DSMB, which is, U01, that's the case, but in other mechanisms, it may not be. A clinical endpoint adjudication committee, I think, is also really important in terms of this probe design of blinded endpoint. So a committee of experts, in our case, radiologists, as well as hematologists, to be able to review PHI redacted source documents, scans, et cetera, to adjudicate an endpoint using standardized internationally accepted definitions. And who's going to manage that? Again, independently from the investigators. That would best be with the Data Coordinating Center. So, we have charter template, SOPs, et cetera, around managing that process. That was handled by the U01 DCC and KIDS. In COVAC TP, that's something we handled here because it was a phase 2, it was not randomized, it was not blinded. Next slide. All right, so I'll go very briefly through the functions of these committees because I think we hit on them already. But I think when institutions deliberately think about establishing expertise and processes for these, we're much better able to support investigators to conduct multi-center trials. And other times we partner with other institutions or for-profit groups to be able to provide certain functions, but it's great to see it when institutions can do so. Next slide. So a steering committee, just very briefly, has responsibility to approve the study design, the analysis plan, to monitor overall and per site performance, review any DSMB or DSMC recommendations, and make final decisions on those. If the sponsor is NIH, it's not the only decision. There's other levels of decision at NIH. But in terms of flow of communication, that's typical. And the membership of the steering committee often includes both clinical and other scientific expertise, and importantly, methodologic expertise, clinical trial experienced biostatisticians, for example, as well as sponsor representatives. In the case of a U, it would be an NHLBI program officer or director. Next slide. Data and Safety Monitoring Committee, I won't go into too long, but here again, the purview is to monitor safety experience, make sure that the study is not futile in terms of it's not accruing, and shares that kind of purview with the steering committee, but focusing especially on safety and integrity, and recommendations about whether to continue the protocol with or without modification or to close the trial. And membership is similar to that which was on the steering committee, but also ideally having bioethics trained member. Next slide. And then I already mentioned the Clinical Endpoint Adjudication Committee, and its expertise really depends on the disease area and the endpoints to be adjudicated. But in the KIDS trial and the COVAC TP trial, it was comprised of radiologists and hematologists predominantly, adjudicating the bleeding safety events and the recurrent VTE efficacy events. Next slide. So quality assurance of endpoints is not uniquely reliant on a CAC, a clinical endpoint adjudication committee, but it's also in the overall protocol design. It is making sure that the endpoints are clinically relevant, that they're standardized, that they're validated, quote unquote, with many different renditions of what it means to be a valid endpoint, that sites are trained on that outcome assessment or measurement. And that there is an adjudication committee in place. So I wanted to make sure that folks didn't take away that the CAC is, that quality of an endpoint relies completely on the CAC. There are other really critical synergistic aspects. Next slide. So in terms of the payoff for KIDS, you see that really slow creeping phase. We could call it the K23 period, and then that steep incline there is the U01 period, and shortly prior there too under a bridge grant. What was exciting for our colleagues is that we were realizing, and this is the largest academically led trial in the disease field of pediatric VTE, so that kept folks engaged and excited. Next slide. Especially when they were not being paid very well until the U01 period. And so ultimately, very recently, January 11th, this came out in JAMA, and there was a really complimentary editorial that I encourage you to look at because it is focusing a lot on the methodology and a rather novel analytic approach called the bivariate endpoint analysis, which the credit goes 100% to John Kittleson, who is a wonderful biostatistician from University of Colorado, who used to run the department there and is now quote unquote retired, but active and still engaged in the U01 and beyond, but also published that method in Statistics in Medicine and some clinically applied publications in other journals. But for those who want to see the formula and the real derivation and proof, it's in Statistics in Medicine from, I believe, 2020. Next slide. So just to quickly orient you, we screened a lot of patients in order to meet eligibility criteria. We had 417 who were randomized, and then we had an additional 100 plus patients who were in the pre-specified parallel cohorts that were continued to be followed in the same manner, but were not randomized. And then in the per protocol analysis, we had approximately 300 patients. Next slide. I won't go through all the demographics, but the median age was 8, and the vast majority of kids were, if you go to the next slide, were had venous thromboembolic events that were in the lower upper extremity. So they were what we called deep vein thrombosis. But given that in kids, a lot of times VTE are cerebral sinovenous thrombosis, or at other sites, sometimes right atrial thrombosis, et cetera, although very few in this trial, we didn't want to exclude those. And you might say, well, gosh, that's a really heterogeneous study population. And yes, it is. It's also really heterogeneous when you consider that, newborns are in the same trial as teenagers and young adults, technically, the 20-year-old who's enrolled. So we stratified randomization by plot location as well as by age group. And those straight to help, but you can still, understand that there may be more work to do in specific subgroups, right, to really see if it's generalizable. And so if we go to the next slide, you see here a comparison of, in the per protocol population, the Kaplan-Meier type curves for the 6-week and 3-month arms with regard to the primary efficacy endpoint, which was symptomatic recurrent venous thromboembolism within 1 year. You see the 12 months there, and you see how closely those two functions approximate each other. And the absolute risk difference for 6 weeks minus 3 months was negative 0.04%. On the next slide, thanks, Taylor, you see the primary safety endpoint, which was clinically relevant bleeding events in the per protocol analysis, minus 0.05% for 6 weeks minus 3 months as within 1 year endpoint. Next slide. And so with John Kittleson's really, I think, elegant way of showing the trade-off between recurrent VTE events and clinically relevant bleeding events, that as clinicians, you're constantly thinking, or as patients, you're constantly thinking about when you are entertaining staying on a blood thinner for X more weeks. You know, it's the trade-off between the bleeding safety and the prevention of recurrent VTE, which is efficacy. And so in this two-dimensional space, you see that the confidence rectangles for that trade-off, that bivariate trade-off for the per protocol population, lie to the south western quadrant of this non-inferiority bound, and the ITT population was even further away from crossing over the non-inferiority bound. So it met our a priori criteria for non-inferiority based on both of these both the per population and the ITT analysis, or the all randomized patient analysis. Next slide. So in terms of COVAC-TP, trying to prevent thromboembolic complications of COVID-19 in hospitalized children during the pandemic, our goal was to look at safety. We enrolled 38 patients. We actually enrolled 40 with 38 meeting criteria for the primary analysis, which was our target. We did that within 1 year across the multicenter study. And we had 0 patients have a primary safety endpoint of clinically relevant bleeding, which met our pre-specified threshold for a preliminary conclusion of safety, that we could go on to an RCT if that was the next feasible step. Our secondary endpoint, we defined 0.5 milligram per kilogram per dose, achieve the targeted anti-10A levels for using enoxaparin for this indication twice daily, which is what was in fact the dose being used clinically when we entertain thromboprophylaxis in children who are hospitalized. So that was nice to be able to substantiate that with some real evidence. And then lastly, our exploratory efficacy endpoint, we had 2 patients who had quote unquote breakthrough VTEs. So as you know, COVID-19 is a really pro-inflammatory prothrombotic illness. And in kids, there's a phenotype called multi-system inflammatory syndrome of children, where kids are really inflamed. And despite these doses of enoxaparin, 2 patients out of the 38 ended up having thromboembolic events. So one of the questions is the dosing too low? And that question is still very active in the adult literature if you've looked at some of the New England Journal and other publications on prevention in COVID-19. Next slide. So to wrap things up, the design of multicenter trials, especially in challenging settings, really needs to look carefully at, are we ready for definitive RCT? Are we even ready for pilot RCT? Should we be doing a cohort study? And in the case of COVAC-TP, you know, were we ready to do a phase 2, or did we need to do something prior to that? We need to leverage institutional infrastructure for CCC and DCC. We need to establish oversight committees, engaging academic experts. It's all kind of hard work and long haul, but the payoff is the opportunity to advance the field and potentially impact care. And with KidsDOT, this is changing the standard of care. We're going to hope to measure that in the next grant of, you know, how is the duration of therapy changing in the field, and what are barriers and facilitators of uptake of the new evidence. But that's the exciting part of being able to do these trials. So appreciate it very much, and I look forward to having questions that folks have. And the next slide just acknowledges one more individual. If you go a little bit further in terms of the mentorship, we're never too far in career to have a mentor. Sometimes they become an advisor. And the late Will Hyatt, who saw this trial all the way through up until the first submitted draft, but unfortunately passed away before the final drafts that were accepted for publication, was a fantastic multi-center trialist mentor for me during my days in Colorado, but also as I was at Hopkins. So thanks again.
Thanks, Neil. Great talk. We are open for questions, and I have several, but so I'm gonna start with one that's very pragmatic. You have an endpoint analysis committee, and you have the clinicians acting upon the data that they get. What kind of time frame did you use for the endpoint analysis? In other words, how fast did they have to make their decision? And what did you do when the clinicians did something that the endpoint analysis committee on judging the endpoint might have said they shouldn't have done? You know, they decided that, you know, this was terrible, and they continued medication where the endpoint committee said, no, it didn't really happen, whatever. I'm just sort of, how did the endpoint, I know how the endpoint committee would be counted in the trial, but in the management and the behavior of the clinicians, how did those data fit in, if at all?
That is such a great point, and it really doesn't come up very often. But we put a lot of thought into that up front, and when we engaged all the investigators, we said we are going to do a central blinded review of these events, but it is not happening in real time. And so it's important that you're comfortable managing your patients as you would before the trial, or if they were not meeting trial eligibility criteria. In other words, if you think that this is a recurrent VTE event in your patient, you're going to manage them according to the standard of care for recurrent VTE. And we are tracking that you investigate, you know, you have an investigator reported recurrent VTE outcome event. But our analysis, our primary analysis and our secondary analysis all depend on the adjudicated events. And so there were, I'm sure, events where there was a recurrent VTE based on local clinical judgment of the radiologist, the hematologist, et cetera, and they restarted anticoagulation. They did a prolonged course because the standard for a second event is to treat longer than the first event. And then if that was adjudicated as not a new event, theoretically, they may have treated it differently locally, but from the beginning, we all had the agreement and buy-in. And even in the informed consent discussions that I had with my patients, I said, the way the trial works is there's another committee looking at these events, but we're going to treat you based as if you weren't on this study, if we think you have a recurrent event. Similarly, if you have a bleeding event, whether or not it meets criteria for the primary endpoint definition for safety, we're going to use our clinical judgment and hold your anticoagulant until your bleeding event has resolved. And we track start and stop dates, et cetera. But the findings were really based on the adjudicated event and not what happened thereafter. It would be really interesting for us to actually do an analysis of what happened after. But what's really interesting, Doug, is that we had so few events in the trial, so that all of our proposed secondary analyses that related to predictors of outcome are really not feasible. You have a couple recurrent VTEs, and you've got a few patients that had bleeds, and so in each arm. And so to me, that also was really satisfying, because clinically it answered the question, even if you didn't buy into the statistical method. If clinically you're seeing so few patients having recurrent VTE events, you really question, why are we treating so long? And some people are going to say the next question is, should we even be treating 6 weeks? Should we, once they're discharged from hospital, should we be stopping anticoagulation and letting them get back to usual life in an obesity epidemic rather than having kids be couch potatoes until they come off blood thinner. So I think all kinds of questions, but to answer it more succinctly, we set up operationally from the beginning. Everyone bought into the notion that you will treat according to your local decision, and you're not even going to know the adjudication until the trial results are unblinded.
And did you have a time frame for the adjudication to occur after the events? In other words, did you say the committee had to decide within X days or X weeks of the event that was in the data, or did you just have them meet periodically and review all the events that came their way?
It was the latter. Because of the fact that we said it was not going to influence patient management, we had the luxury of, meeting quarterly, or if we had a lot of, kind of a flurry of events, we could meet more frequently. But that was also very pragmatic, because as we got into the pandemic, all of the other challenges of people's schedules and, how overwhelmed the clinical folks were in particular, but everybody was, that was really helpful to not have a firm requirement of X days from an event. We had a minimal frequency of the committee meeting each year, tailored to the volume of activity.
Okay. I have a couple more questions, but I'll open it for other people first.
It looks like Larry Moulton has his hand up, or maybe that's just part of his funny avatar.
That's just the funny avatar.
Looks
Okay.
great, though.
I have a question, though. Can we go back to the bivariate analysis? I'm not quite, could you go through it again? I sort of interpreted it exactly the opposite of what you said the conclusion was. So I'm sure I didn't understand it quite right.
Yeah, and I didn't spend a lot of time on it. So, my caveat will be that some of my best colleagues are methodologists, and that I got just enough methodology training to be dangerous during my PhD in Colorado, skewing as much biostats courses as I could. But I am by no means an expert in the bivariate analysis method, and John Kittleson is. But what I will say is that our investigator group and our steering committee got together and defined a non-inferiority boundary based on 3 scenarios of trade-off. If we were to see this excess in bleeding with one of the arms, how much of a reduction in VTE episodes would we tolerate as a trade-off? If we saw this much reduction in bleeding events, how much excess in VTE episodes would we tolerate? And in that informed by judgment more than by operating characteristics, our non-inferiority curve for the trade-off. And then the analysis looked at the absolute risk differences in our primary efficacy and safety endpoints simultaneously in 2 dimensions. Where does the point fall on this curve? And then based on the confidence intervals around those point estimates for the risk difference in both endpoints, we have a confidence rectangle. And we were going to conclude non-inferiority if these completely lied within the blue shaded area, and in terms of risk reduction for primary endpoint events for the 6-week arm when compared to the 3-month arm. And so the red is the per protocol analysis of 300 approximately patients, and the all randomized patients is in blue for 417 or so patients.
Well, what confused me was, it looks to me, if I understand what you said, that the ITT analysis is more favorable in both harm and safety, but I thought you said the opposite.
I meant to say that the ITT was even more conclusive for non-inferiority if you look at the distance from the non-inferiority bound, but they both met criteria because the northeast vertex, or whatever you want to call that, the upper right hand vertex of the confidence interval in neither case touched nor crossed over the bound.
Okay, well, thank you. That's how I understood it, but
Okay.
or what I was thinking it said. So I just probably heard you wrong, but
Yeah.
that was an excellent presentation. Thank you so much. But I'll let someone else ask a question.
Okay.
Well, actually,
Thanks for
I'd
the
like-
clarification. I certainly may have misspoken.
So
Thanks.
I want to follow up on this because this is actually a very interesting concept to me. So if in fact one of the lines of the rectangle stayed in the, south of the non-inferiority margin, but the other one crossed either safety or efficacy, you would be able to conclude that it was non-inferior in terms of either safety or efficacy, but inconclusive as to the other. Is that what you would have ended up doing?
Actually not. We were even more conservative. We said if either one of them, either analysis, if in either analysis we could not conclude non-inferiority, we will say that the trial does not support short duration therapy. So we really put ourselves in a tight spot. And in that way, it was really fortunate that, number one, clinically with such a low event rates, clinicians were going to say, I don't understand your non-inferiority. I don't understand your bivariate. There's so few events. I'm not treating 3 months anymore. But we were really fortunate that not only was it a low event rate, but both analyses lined up in the non-inferiority conclusion. And I think it's a really good point, about the per protocol analysis often being favored in non-inferiority. That's the way I was trained. But you'd be surprised how much debate there was in the reviews we got as we submitted this first to New England and then to JAMA about our choice of, why did you choose hierarchically that the per protocol was first and the ITT was second, and that you had to have both lining up. And so I will acknowledge I'm not the expert in this, and you guys should probably lead the discussion, but I was surprised to see how much debate there was, even among methodological reviewers, and citing different papers as to whether we should have favored ITT or per protocol. I was glad that we therefore said we had to see both line up, because then everybody could agree.
So the second question I have about this is, you said that you just arbitrarily, a committee got together and decided on the sort of distance that you would consider, difference you would consider clinically relevant. So it sounds to me like you picked 2 points, one on each of the axis, and then the curve suddenly spins out from you picking 2 points clinically. Is that what happened?
3 points of trade-off. And, for more detail, the Statistics in Medicine article is really good. That I can, send and follow up. But yes, those 3 were arbitrarily chosen, but it was informed by, clinical, real clinical kind of trade-off decision making. And we didn't rely just on the steering committee, although it's a large steering committee of over 11 individuals. We also brought it to our investigator meetings early on in the study development, and we ultimately had it also reviewed by the Data and Safety Monitoring Board that preceded the NIH DSMB, and then the NIH DSMB re-reviewed it. Now, all of these folks could be wrong, right, in choosing these points, but it ended up where I would say probably over 90 individuals had at least considered these trade-off points between recurrent VTE and bleed that one would kind of clinically judge as being non-inferior.
So other questions from other people?
Doug, this is Jay. I have a question. Neil, very impressive, especially this slide, because I'm a biostatistician, but would it have been better, or could you have chosen high-risk patients for this trial? It would seem it's easier to show non-inferiority if there are going to be very few events, because all of the patients are low risk.
Yeah, it's a good point. I think, in pediatrics, our first step was to do no harm, right? And so we said, if we're going to shorten therapy and cut it in half, a lot of people were nervous and said, no way, we're going to hurt kids because they're going to have recurrent events. And so we really had to convince folks that we picked an a priori low risk group. So your question is really good though, is, so is the next step to go to those who are at moderate risk for recurrent VTE or even at high risk for recurrence? This is still very fresh. We'll see how much uptake there is in the low risk group. But I think there are certain questions of excluded groups, kids with cancer, kids who had pulmonary emboli, kids who had severe underlying thrombophilia or hypercoagulable states. All of those were not included in this population. And we don't know how to treat them other than to treat 3 months or longer until we do that higher risk trial. So it's a great point. And I wanted to quickly, because I see some of my colleagues from the Florida campus, I want to acknowledge Dr. Soche. He is really the PI and ringleader of the COVAC TP. I am the mentor who learned more from him than he learned from me on that trial, and member of his steering committee. And then Dr. Morrison as well on here was one of the steering committee leaders on the COVAC TP trial. And then lastly, Dr. Amankwa, who led the DCC for COVAC TP and also on the steering committee.
Other questions? If not, it's 9:29. Neil, thank you very much. It was a great talk, and we appreciate your giving it to us, and we look forward to interacting with you in the future.
Thanks so much. It was a pleasure to speak with you guys and interact. Thanks so much for those great questions.